So before we get started, I just wanted to kind of briefly mention that I really encourage students to try and ask questions during the discussion after the seminars. We get a lot of questions from faculty, which is great. You all kind of keep the discussion going. But students also contribute to the work that's done in the HPL, so we'd really appreciate hearing from you. So just a little tip in terms of coming up with questions instead of just kind of passively listening to the talk. Try as you're going to maybe set a goal of coming up with at least one question to ask during the discussion. So today's speaker is Armaan Sekhon. I found out today that I've been pronouncing his name incorrectly this whole time. He's a master's student in Walter Herzog's lab. He completed his undergraduate degree in biological sciences in cellular and molecular and microbial biology in 2020. And then his master's is now in biomedical engineering, and his thesis is on using immunofluorescent labeling, which he's going to be talking a bit about today, to kind of explore the mechanisms of residual force enhancement. So without further ado, I'll hand it over to Armaan.
Okay. All right, so I'm going to get started where I always like to start, which is kind of giving a background on where in muscle tissue most of this work happens. So here we have a picture on the top of skeletal muscle made up of a muscle cell. And inside this muscle cell, we have a myofibril, which is a subcellular organelle. Inside of this, we have a sarcomere. And the sarcomere is considered the fundamental unit of contraction. So it's when the sarcomere that contracts and shortens in length that causes the myofibril, the muscle cell, and the skeletal muscle as a whole to contract. Now, this happens when we have the actin filaments and the myosin filaments that are labeled here slide past one another due to cross bridges. I also want to highlight that we have titin here, which is another filament that is kind of in between the myosin filament and the Z disc on the boundary. And it's a spring-like element that provides passive force. We also have a couple of structural elements that will be important later during the talk. So I want to point out alpha-actinin, which is in the Z disc in the boundary of a sarcomere. And we have myomesin, which is in the middle, in the M line of the sarcomere. Now, I mentioned that based on the sliding of the actin and the myosin filaments, you can kind of, that's how sarcomeres produce force. What we can actually do is we can use the amount of overlap between actin and myosin to predict the amount of force that a sarcomere is going to produce, and as we scale up to a whole muscle. So if we have little to increasing amounts of overlap between the actin and the myosin filaments, we'll have forces on the descending limb. Eventually, we'll reach the plateau where we have optimal overlap. And then past that, we start to have other constraints that reduce the amount of cross bridges and reduce the amount of overlap, which causes a decrease in force on the ascending limb. So using the amount of overlap, we can very clearly predict the amount of force that a myofibril produces or that a whole muscle produces. But now I'm going to throw a wrench into that by introducing residual force enhancement. So this is a property that we've noticed in muscle, where say you have a muscle, and what you want to do is you want to activate it isometrically. When you do an isometric activation, you maintain the length of the muscle, but you do a contraction. So it would kind of be like you're holding a basket and someone's throwing apples in. So the more apples they throw in, the more you're contracting, but the length of your muscles stays stationary. When we do an isometric contraction, we produce a specific amount of force. But now compare that to a shorter muscle. What we're going to do here is we're going to activate this one as well. But now because it's shorter, it's going to produce a smaller amount of force. That's what we would expect. Now what happens is, say we try to stretch this muscle while it's activated so that muscle B matches the length of muscle A. What ends up happening is that they're the exact same lengths. But muscle B, when you hold it isometrically, produces more force. And this increase in force is what we call residual force enhancement. So there are a couple of properties of residual force enhancement that we know about. We can use these as kind of clues to figure out where it's coming from, what causes it. So we know that it increases with the magnitude of stretching. We know that it's sensitive to the initial length of a muscle. It's independent of the stretch speed. And most crucially, we notice residual force enhancement everywhere from whole muscle all the way down to myofibrillar preparations. And the significance of this is that it's not due to connective tissue. It's not due to tendons or anything. It must come from a very fundamental unit inside sarcomeres if it's able to exist at this level. And this comes from a couple of studies done in 2008, where they were able to demonstrate that residual force enhancement does in fact happen in myofibrils. So on the second graph here, we have the residual force enhancement stress value, and you'll notice that it's higher than E0, which is the baseline isometric value you would expect. So for this talk, I'm going to break it up into 3 sections. The entire purpose, overarching purpose, I could say, is that we want to use an immunofluorescent antibody system to investigate residual force enhancement. But in order to do that, we have to do a little bit of preliminary work. So first, I want to talk about immunolabeling of alpha-actinin and myomesin, again labeled in this diagram. Then in section 2, I want to switch to talking about labeling of titin in the proximal and distal PEVK segments. And then finally, I'll talk about residual force enhancement and how we can start to characterize titin-actin interactions. A little preface to this talk, I want to say that all of the experiments that we conduct so far have a consistent protocol. There are small modifications, but generally speaking, we use New Zealand white rabbit psoas muscle. And what we do with this muscle is we chemically skin it in order to get individual myofibrils. With these myofibrils, we incubate them with our antibodies. So we have a primary and then a secondary on top of that. And essentially what happens is that these antibodies will bind exactly where you want them to bind. So say you have one that's specific to alpha-actinin, it'll target there. You put the secondary on top of it, and then since the secondary has a fluorescent marker on it, you'll get light shining from that spot. We detected them on an Olympus microscope, and we did them at around 200 magnification. These are the types of images that I will be showing. In these, what you have is a myofibril that's under analysis in between a needle and a cantilever. And you can see the individual sarcomeres, or at least the thick filaments, which are the dark bands of the individual sarcomeres. So the needle's on the this side, and then the nanolevers and the force measurement systems are on the other side. Now, for a little bit of scale, to make sure everyone's understanding what level we're working at, we have a scale bar on the side that represents 10 micrometers. Compared to this, the diameter of a human hair cell is around 50 micrometers. And the smallest thing that the human eye should be able to detect is around 40. Below that, you have red blood cells and white blood cells at around 25 micrometers. And this is just the length. So if you think about the diameter of this thing, it's like 2 or 3 microns. So it's very, very small. On the bottom, we have an image of a fluorescently labeled myofibril. And you can see the bands that I was talking about that kind of glow and show exactly where we want to on the structures that we label. So with that, I'd like to talk a little bit about actin and alpha-actinin and myomesin. Research that's used immunofluorescent antibodies when it's tried to label sarcomeres and myofibrils, it has been conducted in the past. But in research that's been done in the past, there's been kind of a controversy over the methodology to use and the results that you get when you use it. The main one is that when you're putting antibodies into an area that's already very compact, there's already a lot of stuff there. Are you going to prevent the ability of that sarcomere or that myofibril to contract? Are you going to change the dynamics? So in a study in 2006 by Telly, what they noticed is that the sarcomere length was the exact same. It stayed consistent even after they did labeling. The force per cross-sectional area was expected, so they were getting good forces. And the contraction kinetics were relatively consistent with what you would expect. But studies done later on in 2008, and then even here in the HPL in 2015, tried to replicate these methods, and they found anywhere from upwards to a 2 times reduction in the active force that you get from a myofibril. So it was a priority for us to kind of develop a method where we were able to label, but not just label, we wanted to be able to label well, and label by preserving the dynamics of the sarcomeres. Another thing that we wanted to do is we wanted to measure the distances between the M lines and the Z lines very consistently. So usually when you look at sarcomeres under microscopy, it's done with phase contrast microscopy. That's the black and white images like the one above. When we use these images, we have inconsistent banding intensities, asymmetrical striation patterns, and other issues that mean that when we try to measure, say, the length of a specific sarcomere, we have to kind of approximate. And based on whether you do it manually or if you use an algorithm to do it, you can get changes in the length of a sarcomere by 18 to 22 nanometers. So there's an inherent limit to the amount of resolution you can get with these methods. We wanted to kind of supersede that by using immunofluorescent antibodies. So for this section, the priorities that we had were to label alpha-actinin and miomycin, but then to determine if first inserting the antibodies into the sarcomeres compromised the function of those contractile elements. And then we wanted to figure out if even doing this has a point. Are we even getting better resolution with these types of images compared to phase contrast images? And the antibodies were placed on alpha-actinin and miomycin. So for the forces that we got, what we did is we stretched 7 myofibrils from an average length of 2.5 microns to 3.2 microns. What we noticed is that in between controls and labeled samples, we got consistent passive forces in both conditions. And you can see here that mean passive stress values of 60 and 66 were reported in the two conditions. For our repeatability experiments, we wanted to do a study where we would measure the length of a given sarcomere and then measure it again in a following week and then measure it again in a week after. What we wanted to see is that in our phase contrast images, how much variation do we get between those measurements if we try to measure it over and over and over again? Versus when we have these beautiful myofibril preparations that are immunofluorescently labeled, what are the variations in the lengths that we get? Generally, what we found is that, as you would expect, in the immunofluorescently labeled sarcomeres, we were getting less variation, which means we were being more consistent with ourselves or repeatable with ourselves. Here you can see the kind of structures that you would have to label in phase contrast or try to measure out in phase contrast. And again, we have these beautiful lines here to use instead in immunofluorescence. Another thing we could do with this immunofluorescent system is characterize sarcomere lengths and also the amount of non-uniformity that we observed in our sample. So sarcomeres all have different lengths. So you can have a myofibril and the average length can be 2.5, but some sarcomeres on the end will be 2.7, some on the other side will be 2.3, and so it averages out. And this amount of non-uniformity is inherent to all myofibrils. With our immunofluorescent system, what we wanted to do is we wanted to see if we're changing the amount of non-uniformity that we see, and also to characterize it and see how it behaves. So for those types of experiments, we also stretched those 7 myofibrils from 2.5 to 3.2, but then we brought them back down to 2.5, and then back up to 3.2, down to 2.5, and we repeated the process. For non-uniformity, what we wanted to do is we wanted to have an initial position where we measure the individual sarcomere lengths. Then we wanted to go to a position later on and measure those sarcomere lengths again and see if the amount of non-uniformity changed. So are the same long sarcomeres staying long? Are the same short sarcomeres staying short? In our control samples, say we're measuring long position 1 and long position 3, so these are both at 3.2 microns, we have a nice correlation between the two values. So whatever length the sarcomere is at the initial position roughly predicts the length that it's going to be at the final position. This matched for our labeled myofibrils. So the amount of non-uniformity didn't change in any significant way, even with the introduction of our antibodies. Now looking at shorter positions, so here the average is 2.5. We wanted to measure if the same sarcomere stayed short and the same one stayed long. What we noticed is that we had a correlation value of 0.8, an R-squared value of 0.8 in our controls, and it was 0.48 in our labeled, which is still a medium correlation. So we are relatively predicting that the short one stays short and the long one stays short. But what this means is that although the antibodies might not compromise sarcomere function at long positions, at long lengths, maybe when we start to compact the structure and bring everything in there a little bit closer together, the physical space that the antibodies take up starts to become a restriction. So that's kind of how we would explain this. Now, from previous literature, people who noticed a 2 times reduction in force, what they said was that it was a concentration-dependent effect. So we hope that if we reduce the concentrations that we were using for these experiments a little bit more, we would be able to resolve this. Either way, to summarize this section, we thought that immunofluorescent antibodies didn't compromise sarcomere dynamics in passive stretch conditions, that they offered resolution advantages over phase contrast controls, and that sarcomere length distributions remained consistent and non-uniformities were maintained to a comparable degree, except that might not apply for the shorter lengths that I mentioned. So with that, we had the confidence to try labeling something a little bit more invasive. Now, what we wanted to do is we wanted to label a structure that's length changes while a sarcomere contracts. The actin filament and the myosin filaments, they're relatively consistent in length, and what they do is when a sarcomere contracts, they will slide past each other. But titin is a spring-like element, which will crumple up when a sarcomere contracts, and it'll stretch elastically when you elongate it. So we figured it would be a good target for labeling. Titin is a filament, again, as I mentioned, between the Z disc and the thick filament, and it runs actually all along the thick filament as well. In this space, this is kind of an image of what it looks like. We have proximal tandem IG domains. We have distal tandem IG domains, and we have the PEVK region. These are individual domains that can kind of fold up or they can be pulled apart, and they're the ones that produce passive stress that titin is known to be responsible for. When we wanted to label titin, what we wanted to do is we wanted to initially determine if we're compromising the function of the contracting elements. Again, like with the alpha-actin and myosin, but this time, because we're labeling something a little bit more invasive, we wanted to do the same type of control experiments. So we labeled on the proximal and distal side of PEVK. We figured this was a nice spot for labeling because what it would do is it would separate out our entire titin filament into the three distinct regions, the proximal, the PEVK, and the distal. When we were doing our experiments, we stretched myofibrils from 10 myofibrils from an average of 2.4 to 4.4 microns. So we went up incrementally and we waited 30 seconds in each phase, and then we stretched to the next one and we just kept going up. In these positions, we measured force, and based on that force, we came up with these results. So what this shows is that as we increase the length of the sarcomeres, the passive stress that titin is providing, the amount of resistance to the increase in length, is increasing. But what we notice is that it increases at a relatively, the values are pretty consistent with one another across all of our myofibrils. And when we compare to literature, that's also done this type of stuff for unlabeled myofibrils, what we notice is that from this paper in 2015, they produce these types of values. Compared to our values, we have pretty good consistency between the two. So what I'm trying to say here is that our titin labeling is not as invasive as you might expect. It's actually doing exact same thing, or even producing more force as opposed to decreasing the amount of force twofold compared to unlabeled samples. Now, titin has a number of roles inside sarcomeres. It's important for providing a structural framework for the rest of the components in there. It's involved in a lot of cell signaling, so it has different sections that can be phosphorylated or communicate with other enzymes. But most importantly, it's the main determinant of passive tension in stretched, non-activated muscles. So what we wanted to do is we wanted to characterize its mechanical role in unfolding and providing this passive force. I mentioned that titin has three distinct segments, but for this diagram, I'll simplify it to the two. So we have the proximal IG and the PEVK, and you can regard the distal IG for now. But essentially what happens is when titin wants to provide passive stress, first you'll have the IG domains lengthen and align with one another. Following that, you'll have the PEVK start to stretch out. And then if you go to lengths beyond, what'll end up happening is the individual IG domains will start to unclasp and straighten out as well. With our system, what we could do is we could try to characterize this unfolding behavior. So we could look at the type of labeling that happens and try to figure out at what lengths does the PEVK start to unfold? At what lengths does the proximal IG domain unfold until? As I mentioned, the advantage of having our system with a label right before the PEVK and after is that we can separate out into the three distinct regions and we can look at them individually. So a presentation done about 3 weeks ago by Chris Sekhon looked at the proximal tandem IG domains and how they unfolded and the rates of that. So I'm not going to talk too much about that, but I will talk about the PEVK region and what we measured for its unfolding. When we label with these antibodies, these are the types of figures that we produce, where you can see the phase contrast image on the top, but then on the bottom, you can see the proximal PEVK labels in red, and then the distal PEVK labels in green. Essentially, when we wanted to measure the PEVK length, we would subtract the distance between two sets of labels in both the green and then the red, and then subtract them from each other. And then that's how we would get individual PEVK lengths. Tracking sarcomere length versus PEVK length, we found that the PEVK length starts to lengthen at about a sarcomere length of 2.5. And it does so all the way up to around 3.8, after which the PEVK is fully unfolded and the length of it plateaus out. So it's reached its contour length at a value of around just below 0.3 microns or 300 nanometers. Comparing this to literature, what we found is that predictions for the contour length so the fully extended length of the PEVK in human soleus muscle is around 0.28. Comparing to psoas muscle in rat tissue, where they found that the contour length should be a lot higher, but we are within expectations based on literature. So in conclusion for this section, immunofluorescent antibodies don't compromise sarcomere dynamics even when we're labeling titin, and PEVK segments lengthen to around 3.8 when they reach contour lengths, and afterwards the IG domain starts to unfold and we can start to look at the other behaviors. Finally, we can go back to talking about residual force enhancements. So titin can increase its stiffness in response to contractile conditions. This has been noticed in a lot of experiments in the past. Mechanically speaking, there are two proposed mechanisms for how a spring like titin might increase its stiffness, at least in a biological context. One is that titin can change its inherent material properties. In the context of sarcomeres, you could have calcium binding to it, which makes the spring stiffer. The second option is to have the actual spring itself shorten, so the region of it that can still stretch out has to overstretch. And so you produce increased stiffness from that. Now, the reason to look at titin in the context of residual force enhancement comes from experiments done in 2002, where they found that on the descending limb of the force length relationship, you can get residual force enhancement to occur. And this is in part due to the passive elements that are there. So looking at passive elements inside sarcomeres, we logically come to the conclusion that we need to take a closer look at titin. Between the two explanations, whether it's calcium-based increase in stiffness or a titin-actin increase, we wanted to look at titin-actin increases for two reasons. The first is that calcium-based stiffness increases alone don't necessarily explain the increase in force in residual force enhancement. You do get a stiffer spring due to calcium, but it's just not enough. The second is that there are experiments that have been done in the past that have shown that titin does interact with actin. So this experiment here is an actin-titin motility assay. Essentially, you have a surface, and on top of it, you have myosin heads. And then you take actin filament and you try to slide them past on top of it. What they noticed is that filaments that had titin attached to them, when you had titin also in the bed of myosin on the surface, the actin would slide across at a slower and slower speed, indicating that there's probably a chance that the titin that's embedded in the surface is slowing down the actin by binding to it. In the context of residual force enhancement, how might titin bind to actin, and what would we kind of expect the spring to do in response to that? Well, in this figure here, we have an image of a regular myofibril that's just passively stretching. So when it passively stretches, the proximal, the PEVK, and the end filaments all lengthen. So you have a free spring. In the context of actin-titin binding, what would happen is that you would have a binding site between titin and actin, and it would restrict the early positions of the spring from lengthening. And so the free spring that's left would have to overstretch to compensate to get you to the same lengths. We wanted to investigate this. So we wanted to measure this displacement of different segments relative to one another during our micromanipulation to characterize these titin-actin interactions that might be happening. We wanted to figure out where they're happening and how significant they are during residual force enhancement. So in the context of that, we had labels on the proximal and distal end of PEVK. Now, when you do a free spring extension, what you would expect is that the red label would move a little bit and the green label would move. And you would produce a graph kind of like this. So the position of your red label is increasing because the stuff that's before the PEVK, the proximal IG domains, are increasing in length. And then the stuff in between, and then the green label would move because the stuff in between would also change position or stretch out. In the context of residual force enhancement, though, the red label would be immobilized. Its position would be restricted because there's binding close to it. Whereas the green one would be allowed to move and in fact would have to over move and overstretch to get to the same sarcomere length. So you would expect to graph something like this, where the red one is either consistent or it changes very slightly, but the green label starts to go a lot higher. What did we find in our experiments? So looking at, we have 4 sarcomeres, 2 myofibrils so far collected for this data. But what we found is that in the passive stretch conditions, so looking at the red and the green, the first of the two, what we noticed is that they're relatively consistent with one another, and they do separate out, and the green one is a little bit further than the red one. But when we do an active stretch, the position of the red label doesn't move compared to the passive stretch or active stretch state, but the green label does end up overstretching, so its position is a lot higher than it would be in the comparable passive stretch. Now, another question we can ask is what might cause these titin-actin interactions? There's been a lot of theories that have been proposed, but we do know that it needs to be under calcium-based activating conditions. And that's the only situation we get residual force enhancement in. So maybe we can take a closer look at calcium itself. In these experiments done in 2018, what they found is that when you have an increased amount of calcium, you will have more precipitation of N2A region of titin and actin together. So essentially what it's saying is that an increase of calcium causes these two things to bind together. And so in a co-sedimentation assay, they'll appear more frequently. So the idea here is that maybe it's calcium. Maybe all you need is calcium, and then that'll cause titin to bind to actin, and you'll get residual force enhancement. Well, to check that, what we wanted to do is we wanted to determine whether calcium-based activating conditions alone, without the presence of cross bridges, would induce these titin-actin interactions, and we would see the same type of residual force enhancement data that I showed you just before. To inhibit cross bridges, we used BDM inhibitor, and we ran a protocol where we stretched 2 myofibrils, again, from 2.5 to 3.6 incrementally. So we waited 30 seconds during each stage, but we gathered data points for all the individual lengths up until 3.6. What we found is that the PEVK length, whether it be in the BDM alone, so without any calcium, or with BDM in the presence of calcium, in both conditions, it's very comparable to one another. And what this means is that while you would expect in a residual force enhancement experiment for the calcium or for the actin-titin binding to cause PEVK to overstretch, it's not overstretching here. You can put calcium into the myofibril, you can put it into the muscle. It's not going to cause, that alone is not going to cause PEVK to overstretch. So you're not getting residual force enhancement. We can look at the increase in stress that we get from calcium being in the solution, from it causing kind of an increase in the stiffness of titin. And we do notice that the calcium is causing our force to increase when we passively stretch our myofibril. It's causing the increase to go up faster. But this alone doesn't reach the levels that are comparable for a residual force enhancement experiment, where you would expect values to be 200 and upwards for that type of force. So to kind of continue this study, I'm going to collect more data for the final 2 experiments that I mentioned. And I'm also going to do a little bit of immunoelectron microscopy labeling to kind of accurately figure out where the labels are. But in conclusion for this section, preliminary results indicate that PEVK is disproportionately elongating during residual force enhancement, and that calcium-based activating conditions alone cannot induce titin-actin interactions or recreate residual force enhancement phenomenon. And with that, I would like to thank everyone that's helped me and open up to questions.
Thanks for the presentation. Really nice photos as always. First one is, I know this non-uniform sarcomere kind of distribution is well known, but I guess maybe I've forgotten, but what's the reason that this occurs? Like, is there utility to the distribution being non-uniform?
For utility, I'm not sure, but the reason it occurs is because you can have different isoforms of titin within each individual sarcomere. So you can have longer variants, or you can have shorter ones, and then those stack up to cause distributions or changes into the lengths. or that variation that you see.
I have a bit of a late question. So the nano levers that you use to measure the force, how exactly do those work?
Okay. Essentially what we have is two nanolevers, and what will happen is that the myofibril will be attached to one of those two. So when we try to activate our myofibril, it'll start pulling on one of the two nanolevers, and because we know the stiffness of the two, and based on the distance that they start to separate that we can measure, we start to know how much force is being produced that's pulling on one of the two cantilevers. So with that, we characterize force. Yeah.
I don't think I understood the contour length of the PEPK region that you found in comparison to the human soleus, and then your comment about the rabbit psoas. Could you just comment on that?
Right. Yes. So what I wanted to say about that is that the so PVK segment has around 800 residues in psoas muscle, and each of these, when it's fully extended, has a given length, which is I think it's 0.36 nanometers. So if you multiply those two together, you should get the maximum amount of length that an unfolded PVK segment can produce, which is I think it's 288 nanometers is what you would expect. Within our data, we were pretty consistent with that value. So although there's a little bit of variation, so a lot of the dots go above and below, which is to be expected when you're trying to do such small work with myofibrils, our trend line still follows this pretty closely. In the experiments that I mentioned that were done in previous literature, the first one was done in human soleus muscle, and the second one was done in rat psoas muscle. Essentially what they did is they did the exact same types of experiments that I mentioned here, but with immunoelectron microscopy. So they labeled and they stretched to whatever given length, but then they had to freeze it, they had to process it for electron microscopy, take it over, and then image it. And those curves are essentially just what they got. So the first study, the one on the soleus muscle, predicted a contour length of around 300 as well, but then they got values above that, and as did the rat psoas muscle as well.
Did you look at the active force in the actinin and myosin labeled? I don't think you showed that.
Not for the, we didn't look for active force in the, or like we did experiments and we got forces, but not enough to produce any figures. What I can say about that is that it was relatively consistent with what we would expect for unlabeled stuff. For the residual force enhancement experiments that I'm doing right now, to do a residual force enhancement experiment, you have to activate isometrically, and then you relax it, and then you activate at a shorter length, and then you pull it to the long length. And the activations that we're getting for the Titan labels in these residual force enhancement experiments are upwards of 200 and 300 nanonewtons per micrometer squared, which is very good. It's showing that the myofibrils are behaving as expected.
So why is it known why the PEPK region elongates before the distal IG domain? What's the sequencing of the events and why does it occur this way?
Specific sequence, I don't know, but the PEPK stands for the proline, glutamate, valine, and leucine PEPK. Yeah. So it's based on the amino acids, and essentially that's just the structure of it itself. From experiments where they try to pull on the individual IG domains or pull on the segments of the PEPK, they just found that the PEPK, based on its structure and the way it's composed and the amino acid residues that it's made up of, it just has more resistance. So it's considered a high stiffness spring element, whereas the IG domains are the low stiffness element.
I noticed that you were saying unfolding of PEPK. Like, is this meant to intentionally say unfolding with what we know of the meaning of unfolding, and you believe it's an unfolding of the PEPK?
I am not sure. It's, yeah.
How long do you do your myofibril preps last? So you said you did like repeated measures. Is that on the same myofibrils? Like were you looking at the consistency of circular lengths over time?
No, so those ones we collect the data sets, we take the image, and then we analyze it for a first pass, and then a week later for the second pass, and then a week later for the third pass. these individual preparations, when we have them on the microscope and mounted and everything, they'll last for maybe an hour, maybe an hour and a half, if you have a really good one.
I might have missed it, and this might be kind of a stupid question, but does the, you tested to make sure that the passive force between unlabeled and labeled is very similar when you do the alpha actinin and the myomecin labels. Did you do that also for the PBK as well?
No, I don't have that data. So just the controls that you would need to run to.
What do you think? Would you think would, now that you're sort of in the guts of the myofibril I-band region, would you expect any difference?
It doesn't seem to be different from stuff that you guys did in 2015 on the same myofibrils, but with a little bit different solutions. It seems relatively consistent. And if it's consistent within activation experiments and residual force enhancement experiments, I can't imagine that in just passive conditions alone, which are a lot simpler as experiments, that it would be any different. It seems that we're getting good labeling. As for why, I can only speculate. So in 2006, when they tried to label Sarcomeres, label myofibrils with antibodies, they included blocking solutions like BDM, sorry, not BDM, bovine, albumin, BSA, and things like that. But in 2008 and 2015, they didn't include that stuff. And when I didn't include it, what I noticed is that my myofibrils were very sticky and that they would try to stick to the cover slip, so I wouldn't be able to pick them up and attach them to the needle or the cantilever. By including it, I was able to pick them up. And what would happen is that they wouldn't be damaged when I had them mounted. So if they're sticky, then when you try to peel them off, you can start to see a little bit of fraying in the myofibrils. Whereas I didn't notice that after I started using the BSA. And that's what I attribute to the high, the consistency of the results. The other thing to mention is that we do use, newer cameras and there's advancements and stuff, and we use way, way smaller concentrations. And I'm sure that has an effect on the amount of antibodies that end up in your sarcomeres. So it comes across in the data as well.
Just wanted to ask if you could show your conclusion slide again, because I thought it was a little bit fast for me. And then let me just make sure I understand. So just in layman's terms, I gather that you're not enthusiastic about calcium interactions being the explanation for the, okay.
I do believe that they happen, but I just don't believe that, that they do increase the stiffness of titin, but I just don't believe that it's enough to cause the forces that we see in RFE. And I don't believe it's enough to cause titin binding to actin. I believe that there has to be something else involved, like cross bridges and forces starting to pull on everything that allow for those interactions to happen. And then that adds the additional force that explains why residual force enhancement producing force is so high.
Okay, and then you are enthusiastic about titin actin binding being a contributor to this stiffness.
Yeah.
And when you say upstream of PBK, what does upstream mean?
In the kind of just in the diagram of titin, you would have to imagine that there's a binding site occurring somewhere here between titin and actin for a titin-actin interaction. But the location of this binding is kind of contested. There's a lot of people that say that it binds in the middle of the PEVK region, in which case you would expect no change in the position of the N2A label following this binding, but you would see changes in the position of the green label following this binding. And then there's some people that say it binds in the region right before it, so right before the N2A where I'm labeling. And I don't feel like I have enough data to say exactly where it's binding right now, but based on the amount of movement that I do see in the red label, even after you do activations and residual force enhancement, I want to say it's happening before. But again, I don't have enough data to conclusively say that, but I'm just speculating.
Okay, so before means upstream, which means to the left in the picture.
Yeah,
right?
yeah.
Thanks.
We go to the basic picture with the acting and myosin.
Okay. This one?
The one where you have to type in stretch or
Oh, I'm sorry, which picture?
have to tighten, stretch for a while, and then it comes down.
Okay, yeah. So you're talking about, is it this one, right?
Yeah, wait, this one. So you have the point in the lower left part of the picture where the titan is fixed somehow and then not fixed anymore, correct?
Yeah. So in a
Now, let's assume you do an experiment. Is the point where you fix always the same element of titan where the thing breaks?
I would say that the
Does it move around?
I would say that the point on Titan would be consistent, but on Actin, I'm not sure about whether the point is the same.
So it's always the same on titan.
I think it's the same point on Titan, yeah.
How do you know that?
Well, I wouldn't know based on this data. The residual force enhancement experiments, we need to collect a little bit more, but the reason I say that it should be a consistent location is because there's very specific residues that need to be there for like interactions with phosphorylation or with other enzymes and stuff. And each location has a specific binding interaction. So for a titan actin interaction to happen, I would just imagine that it's a very specific residue, specific structure there at one specific location that's causing it.
So the titan has to know where to go.
Yes.
But the actin just waits.
The length of the actin filament is not going to change very much. It's a pretty conserved structure. So I'm going to imagine that it's pretty much the same. It just stays there.
And where do the commands come from? How is the whole thing controlled?
Well, that comes into the whether is it just calcium there or is there ATP there that's causing it, or are there other things that cause it to happen? We know that it's something that's inherent to our myofibril preparations that are free from all like there's no cell signaling thing coming from like the mitochondria or something. That's all removed.
So if I tell you I want that point at the bottom, the actin of that move 10 centimeters right, whatever that is, it measures. You would know what to do. 10 centimeters right.
I would propose that it can't move 10 centimeters to the right. I'm not sure though. I'd have to go back into the literature and look into it.
It can move to the right.
I would say no. I don't think
Can
so.
move to the left.
No, I'm saying that the position is fixed, that it binds to a
Oh, that's fixed. It's either on or off.
like there's a specific location for that binding interaction to occur. Because on Titan, it's a specific residue, and on Actin, I'm imagining it's another specific residue, and they just have to be there for the interaction
So
to
the
happen.
contribution of the left spring is always constant,
Yes.
and the right spring is not constant.
Yeah.
So if you would have had to construct a tight element, would you have constructions like that?
I think it gives Titan a couple of functions that add on to its ability to just be a passive spring in Sarcomeres. As for what those functions allow it to do, I'm not sure. Maybe it helps Titan, or it helps the entire sarcomeric structure be a little bit stronger and a little bit more resistant to kind of very damaging stretches while you have an activated muscle. So say doing an eccentric contraction or something, it'll keep the integrity. But specifically, I don't know.
But if that point, that contact point is not a contact point, I have a completely different situation force-wise.
Yeah.
That's clever.
Yeah, I think it gives Titan more functions. And as for what these functions allow it to do, I feel like there's not been a lot of kind kind of work dedicated to that. Mostly it's been right now trying to figure out exactly what this interaction is.
So I know that in insect muscles, they have kind of this prototitin molecule, if I'm remembering correctly. So is there an idea about when titin evolved in the kind of evolutionary history? And is there some specific association, with the development of it, or is it just
I'm not
totally
sure.
random or something?
I mean, if it's in invertebrate muscles as well, I would have to imagine it's very early, but I don't know. Yeah.
Has anybody seen that? I mean, if I look in a microscope, do I see that?
It would be too small to see. And the other issue,
Why do we know that it is like that?
based on data on passive force enhancement. So passive force enhancement is a phenomenon that occurs after residual force enhancement. So in residual force enhancement, you get higher force than you would expect during an isometric contraction. You have that elevated force. But what happens is that when you eventually deactivate the muscle, we notice that there can still be this increased amount of force, and we call it passive force enhancement. If it's increased and we've already kind of deactivated the muscle and taken away all the force coming from the active components, the only passive element that we can really examine is titin. So we decided to look there.
So that is basically one model that can explain the results that you have.
Yeah. There are other theories that explain that try to explain residual force enhancement. I know a popular one is the sarcomere length non-uniformity theory, essentially saying that there are sarcomeres of different lengths, and during residual force enhancement, you have the long ones kind of overstretch and pop, and then they start to produce passive force very quickly, and it increases very fast, and then that's where the extra force is coming from. There are a couple of problems just mathematically with that hypothesis. One of them being that in residual force enhancement, you can get forces that are higher than the plateau, than the maximum amount you would expect for the plateau of a force length relationship. And you would only get that if there was something else that were contributing to force, and it's not just individual sarcomeres that are being distributed differently to produce a more optimal amount of force. But there are other theories out there that kind of try to explain this.
Well, I got an online question for you, Armaan. The question is, for force enhancement experiment with labels, why did you choose to label N2A region and the junction of PEVK and the distal LG domain, not other region and type? Do you think it is N2A region that interact with actin filament during active leptin? And do you plan to do more experiments which is the same that you did with more spots to be labeled?
Yeah, I think it would be convenient to have even more in there so that we can track even more lengths and get a better idea of what's going on. And some of that work has been done, but you do have to consider that with more stuff in there, you are eventually going to start reaching the threshold where you will start interfering with proteins. So starting off with 2 was kind of a good idea for us. We figured it was enough to be informative, but not invasive. As for the question of why we labeled those regions specifically, again, it just comes back to the fact that we wanted to separate out the proximal IG domains, the PEVK, and the distal IG domains. And by labeling at those specific junctions, we were able to separate them out. So it ended up being convenient for that reason.
Come back to the N2A region and why it's believed it's the region that interacts with actin. I think it's based on some NDM, which might not be traded. And then the other thing I wanted to ask you about is the co-sedimentation experiments. Because you say you mentioned you have on the gel there N2A, and when I don't remember exactly what it is, I'm not sure if you do remember. Is it construct N2A and they found it interacts with actin, or is it a titan, an N2A titan isoform, the whole thing, but it's labeled N2A because it's from a skeletal vessel? Remember?
It is a N2A construct with with actin filaments. So it's not the whole thing.
Okay, so this answers Benno's question that's why at least like there is one experiment that supports
Yeah.
N2A and actin interaction.
Yeah, at a specific. Yeah. So yeah, here with increasing calcium, you start to have more precipitation of the two together, which means that this specific region starts to bind to titin. This specific region of titin starts to bind to actin more and more and more with more calcium.
But this contradicts your result that the calcium.
It does. Yeah, I mean, calcium can do a whole bunch of funky things in muscle. This may be one of the things, so it might help with residual force enhancement. It's just based on what we have. We don't think it's the sole contributor, sole cause of residual force enhancement.
That's just going on that. What happens to force when you stretch a myofibril passively? When you stretch it in the presence of calcium, that you inhibit cross bridges?
Well, eventually you're going to pull it apart, but before that, you'll start to.
In terms of force.
In force?
What's the difference in force?
Force will continue to rise upwards to like.
I mean, you pull passively and you pull actively with calcium, but you inhibit cross bridges. What's the difference in force?
The difference in force was about 50 nanonewtons per micrometer squared. Up to around 3.6 is where we stopped for this experiment.
So it's very small.
It's very small, yeah.
Because when Tim and Venus have done similar experiments, and then Davide did similar experiments with calcium inhibiting cross bridges, the difference is really tiny. In fact, in some of the experiments, we really didn't find the difference. So you can flood the system with an activating solution with calcium. If you don't allow cross bridges to attach, there seems to be something happening. That seems to be at the limit of resolution. But not really a lot. That goes back a little bit to the co-sedimentation experiments, because people have also done, like you showed, the motility assays, where when you add part of titin, then they see that the actin that's crawling on the myosin cross-bridge heads is slowed down. And they have taken that as an interaction, and that it might be explaining residual force enhancement. And my thing has always been, no, to me it shows exactly the opposite. It shows that we cannot explain it. And the reason I'm saying that is because actin is still able to move in the presence of titin and N2A, despite the fact that it's pulled by 2 or 3 or 4 heads. So you're exerting 4, 5, 6, 8 piconewtons of force on the actin and still moving. That suggests to me that if there is a binding and it's therefore slowed down, that binding breaks in the single piconewton range. But in order to explain the force enhancement that Venus and also Tim have seen on single sarcomeres and single myofibrils, we need to have forces on titin up to 200 piconewtons. And if that's caused by a titin-actin interaction, that bond needs to withstand those type of forces. And so I know that everybody has interpreted those experiments as showing, oh, it's interacting, oh, that's the cause. And I always have said, no, that actually shows that the force is so little that it probably cannot explain it. So I always look at that opposite to the rest of the world. But I'm not saying I'm right, but that's just my logic there. So I think maybe another thing to come back to Benno's, you have to be very careful. And I know there's a lot of people saying that titin interacts with actin, and I think it's fair to say that we have done more work in this area than anybody else, than maybe the rest of the world combined. But I'm actually still not sure if that happens. I still see that as a theory. I think to me it's a very appealing theory and a very simple one, and one that would work nicely and beautifully. But at the end of the day, as you mentioned, has it seen an electron microscope, or has anybody seen it? No, nobody's really seen it. So it's still fairly indirect. it's indirect evidence. I would argue it might be fairly compelling evidence. But, you know, would I bet my life on that titan interacts with actin? Definitely not. Would I bet a month's salary? Probably not. You know, I'm too cheap for that. So there's a lot of other things that can happen. So you know, that's kind of like my thinking, our thinking of everything. But I think we have to be very careful. And I know, like just at the European Muscle Conference, I mean, there's people that don't even argue about it anymore. Oh, it binds, we know it, but actually we don't. You know, so we have to be. I think we have to be a tiny bit careful about that. So the other thing then is, if you go back to that Titan slide where it just was with Benno, you know, the question becomes, you know, in 2002, Tim and I predicted that this might be happening, you know, that it might be binding. And then you go, well, why would you ever have that idea? Because we know Titan gets stiffer, but why should you do it that way? And I think that there's several things that went through our mind, and Tim and I discussed that in 2002 when we saw the passive force enhancement. The first thing is that Titan binds to actin naturally and persistently at the very end, you know, about 50 nanometers away before it goes into the Z band, it binds there. So we know there's a permanent binding site there. And then you go, okay, there's a permanent binding site there. Maybe there's some transient binding site as well, because it does already interact with in a permanent fashion. So that doesn't really help because it's always there passively or actively if you stretch or not. And then the second thing is also, you know, Michael was referring to like the evolutionary precursors. And so in some of the invertebrates, there is this, what people consider this Titan precursor called twitchin. And we know that twitchin binds, makes exactly that type of a bridge that you're showing there between actin and myosin. And we know it's regulated by phosphorylation. In the dephosphorylated state, you have that. In the unphosphorylated state, you have no binding. And the only difference is that in twitchin, the binding is very long. You know, it attaches and then it stays on there for hours until phosphorylation, dephosphorylation does it. So there's an evolutionary precursor in mollusks, for example, and in crab muscles where we know exactly this happens, except not on a time scale, you know, like being attached 100 milliseconds and detaching that type of thing. So I think that was, you know, partly the thinking of why we came there, but you know, that doesn't mean it binds. But that's there. But let's chat about something different here. You know, we showed a slide. It's the non-uniformity of the sarcomeres, the testing that we did at the short lengths.
Yeah, that's pretty.
And you were arguing that they look, yeah, those ones, they look
These
similar.
ones.
The R squared value is fairly different. And when I look at the range, you know, then on the left there in the control, it goes from about 2.1 microns to maybe about 3 or 3.1. And in the short ones, from about 2.2 to 2.8 or 2.9. So it's a little bit compressed. And it doesn't seem to be quite.
It is
nice, you know?
Yeah. If you look at the spread, right?
Yeah.
Yeah.
So I wonder if not something might have happened there compared to the other one, because it does look different to me, just qualitatively, and then the numbers seem to be a little bit different as well.
As for spread, it's possible to attribute that to just the fact that in our labeled samples, we were able to measure more accurately. So say you have a sarcomere and its length is supposed to be 2.7. When you're doing a control measurement in our phase contrast images, you might measure it and your number you get is probably 2.9 or 3, and you're just slightly inaccurate. Whereas in the labeled sample, you're exactly on the dot. So even though in both of these samples, we tried to bring the mean sarcomere length to 2.5, so most of them should be around it, maybe we have more spread in our controls and less spread in our measurements and our labels, and that's why it looks a little bit more clustered there.
And they just explain to me slowly, because these are results that I also see for the first time. I haven't seen that. But when you do the experiment, you know, with the two labels, and you do it with activation and BDN, and then you do it active without the BDM, you get that difference. Explain to me kind of step for step, what do you think might be happening there?
What might be happening?
So in the active and BDM first.
So for those experiments, what we do is we essentially we have a myofibril and we put BDM into it, and then we take it from a length of 2 point. Actually, I have a diagram for this. So we have a myofibril.
I know what you did. I'm actually interested in the difference in the results.
Well,
So if you then go to the result.
there's no difference in the results, is what I'm saying. So
exactly. So here, there is no.
There's no difference. So I'm saying it makes no effect.
Yeah.
Whether you have calcium or not.
Yeah. So this is essentially then really a passive stretch and an active stretch with cross-stitch is inhibited.
It's a passive stretch, but everything that needs to be there for a activation is there. It's just that none of those things that you added alone are enough to cause the activation. You do need the cross bridges to be freed, and that's the last missing thing that will start to cause residual force enhancement. And that's when you'll see PVK lengths be different from one another. That's when you'll see forces start to go really high in the residual force enhancement state. I'm saying that in our calcium condition, we have everything but the cross bridges, and we still don't get a residual force enhancement. So I'm saying that the cross bridges are important somehow.
Exactly, yeah. And that's, when I say activation, I don't mean cross bridges attached. I'm talking about it, like the physiologist. So to me, activation means calcium.
Yeah.
So calcium is there, so you have it activated in one case, and then not activated in another case, and you essentially get the same. So then what you measure is PVK. It's essentially the same. So you assume that whatever happens on Titan is about the same.
Yeah.
And so now you do that in the active one.
Well,
Then you also have an experiment where you didn't inhibit the cross bridges.
yes, but for that one, we have just this figure for it. I do know the forces, but
I'm not interested in the forces. I'm actually interested in you explaining to me what happens there. What do you see there?
well,
Just a bit fast for me.
what we would expect is that because we have binding, one of the labels is going to be a lot more stationary. In this case, our red label, which is closer to the binding site. And our green one, because it's in the free spring, and now because the free spring has to overstretch, the green one moves a lot more. And that is kind of what we see here. Although it is a small data set, we see that in our active stretch condition, so in the residual force enhancement stretch condition, where we do have we propose we might have a binding site, we get a bigger force, we get a bigger displacement from the Z disc, so it's moving a lot more. So we do see the PVK length is going to be, to measure PVK length in this instance, what you would do is you would subtract the green versus this green versus the first red, and then this one versus the other one. And what you're going to get is you're going to get a bigger length in your active condition than you are in your passive condition. Whereas here,
go back there, because that is exactly how I interpreted it. That the red, so the second and the fourth one are far apart, so that implies a big stretching of the PVK. But then I look at the first and the third, and I would have expected that they stretch as well. Yeah,
there should be a little bit of a gap there. We didn't detect it here, but it's a pretty small data set.
Okay, so you think there's more data that that might come out because from where to where did you stretch here? These experiments?
This was from 2.7 to 3.2.
3.2.
Yeah.
So you would expect about half of PVK maybe to elongate. So you would expect about 140 nanometers or so, or 150 nanometers.
Yeah.
Which might be tough with the resolution. And then the other one there, I look the medians, you go from about 700 to about 900, so you get about 200 increase. Yeah. But those are two specimens you said, or?
Yeah, 2 myofibrils, and then the individual measurements from 4 different sarcomeres. to get these PEVK lengths or these label positions.
So then the argument being, you have the bigger PVK stretch, because if I stretch the same amount, if I stretch the half sarcomere by the same amount, now more needs to be taken up by PVK than in the other situation where it doesn't bind.
Yep.
So, so then coming back, to the order of unfolding, we heard a little bit Chris about this morning, but the order of unfolding has kind of been that PVK elongates first, and then only at 3.5, immunoglobulin domains would be unfolding.
Yep.
And if indeed that was correct, then you would actually assume that there shouldn't really be a difference, because the immunoglobulin domains up to 3.2 don't really do anything anyway. But I guess Chris this morning in the room meeting showed that that's probably not correct, that immunoglobulin domains probably play a role already at the low sarcomere lengths. Would you then agree that these results support that?
So could you say that again about the proximal IG domains? You're saying
what I'm saying is that wisdom so far, now Henk Ronsier, the experiments that he did, that would suggest, and also Siegfried Labeit, that immunoglobulin domains don't really unfold. They don't really do anything below about 3.5 microns of sarcomere length.
they might,
And since you only stretch to 3.2, if that's indeed true and they don't contribute to the elongation, then you would expect that in the passive and active, all will be taken up by PVK, and therefore the difference should be the same. But according to these preliminary results, it's not.
Yep.
So therefore, that would also not only indicate that maybe there is some binding there or there's some shortening there, but also that proximal segment would also contribute to the elongation in the passive state at very short sarcomere lengths. Because if it didn't contribute, then it wouldn't really matter if it binds or not. Does that make sense?
Yep. Yep. Yeah, I agree.
So what do you think is happening?
I think it's binding to actin. I think that the lack of movement of the red label is just very indicative of that. And yeah.
And then going back to Benno's question, and I think I would agree with you, since items are relatively, has different reasons, different things, that maybe there is a point. So you say you would speculate that the binding site at the moment, I mean, we don't know that, but you would speculate it's closer to the Z line, proximal to the N2A label.
So if it comes in the middle of the PVK region, you would expect that both the reds in the passive stretch or the active stretch condition are going to be the exact same length, because the binding site happens before them, so they're immobilized. If the binding site happens before, then the labels are after, which means that the labels partially should, one or two of them should elongate a little bit. And I mean, this is just 2 data sets, but we do see elongation. So I would hypothesize that the binding site is before, but like very, very close to the red label, just slightly before.
So if you stay with that slide there, then you stay with that slide.
So,
and
yeah.
Right now you show that it binds exactly where you have the one label. So if that's indeed the case, then you would expect that that label
is going to
never really moves.
Yeah.
But if it was more proximal,
closer to the Z line, then you would expect like a very slight amount. Maybe it might not even be detectable by the system, but you would expect a little bit of movement.
But that would actually be interesting, because if it was proximal to both labels, then how far the first and the second label move should give you actually purely geometrically, and from what we know about the amino acids and how long they are and so on, you should be able to pinpoint fairly accurately where it might be.
Yeah, you would be able to back calculate it.
You would agree with that?
Yeah, and I believe that was done a little bit by Mike Duvall's work, right? He was trying to back calculate it as well.
Well, he thought it was a different mechanism.
Yeah.
But then we tried to kind of, because that's what he saw. You know, he had that label. Well, he had the green label there, the one on this lane, the F146. And then
he had one in the middle, right? He never had one on the proximal end.
he never had one on the proximal end, so he never could do that problem. But you could potentially.
Yeah.
If it's more proximal.
Yeah.
I've got a really basic question. Can't you just put 100 labels on and stretch?
Yeah. And upwards of 5 or 6 has been done in the immunoelectron microscopy results I talked about. Like, for this stuff, because it's live myofibrils, we wanted to start off with 2. And provided we're able to get, good forces, good contraction kinetics and everything, then we'll start to upscale it. Because I am interested in maybe putting one very close to the Z line, just like at the very, very beginning parts of Titan. And then that way you do isolate out the individual segments. Like you would get the proximal one, you would get the PBK and the distal, but you would just get them with more accuracy. You would have a little bit better measurement. So I would like to go into that.
But what's the problem putting 6 labels on there?
You can, well, it's like, so an antibody essentially has 2 variable regions. Those are kind of like the parts sticking out of the Y shape. And then those are the things that'll bind to stuff. And what can happen is that the more and more antibody you have, you'll start to get antibodies where one of them will bind to one Titan filament with one of its arms and another Titan filament with another arm. And now suddenly you have them kind of connected. And then that starts to cause issues. And the more antibodies you start to introduce, the more cross-linking you'll start to have.
The other problem I would assume is also that the fluorescence starts overlapping and it becomes hard.
That is
hard to decide which label, where does one label end and where does it start?
Yeah. So right now, the length of the individual bands are about 200 to 300 nanometers. And so we measure the centroid of that and we assume that to be where the signal is coming from. And that's just, we can't detect any more than that because it's the resolution limit of light that we're up against. So we have bands this thick. So if we start to put a lot of bands in there, then their signals are going to start overlapping and we won't be able to see individual ones. The other issue is that if the bands overlap, we need them to, like the red and the green labels like this, because the PVK is a pretty short element. It's about 100 or 150 or 200 nanometers in length. That means that these red labels and these green labels, if you were to take an image of both of them at the same time, they would be right on top of each other and you wouldn't be able to detect them. So for us, it's very important that we have a distinct color for each one so that we can use different wavelengths of light to collect these each individual image. If you want to do 6 or 7 labels, you're essentially just working within the color spectrum. And so you can have green labels and you can have blue labels and stuff, but you only have a certain amount of different colors of light before one wavelength of light starts lighting up multiple ones. So you have to consider kind of, yeah.
Has anybody ever taken a few brilliant mechanical engineers and tell them, if you would have to solve these problems, show me your design?
I believe so. I think a lot of the mathematical models that try to simulate what's happening here are done with help from like mathematicians and engineers and stuff. For me, I'm background in biological sciences, so I like looking at this stuff more than the mathematical side, but yeah. It's definitely something we consider.
The whole thing seems to be an optimization problem.
You're talking about just how this might happen?
Seems to be an optimization problem. You have to be able to achieve certain things with some objective functions. And you can do that different ways. You know what? So why does the Titan on the left hand side be right where the active is? Can it be further down?
I see what you're saying. You're saying
for me that this doesn't seem like an elegant solution.
You're saying that based on where it binds, you would have a free spring, and then you would be able to calculate kind of how much force it should produce to contribute to residual force enhancement. So To that, I would say I don't think we know enough about the way that the amount of stiffness of the individual titin residue, the PVK residues or the distal IG residues to be able to say that based on this binding site, the rest of the PVK or the rest of titin should produce this amount of force. So we can't back calculate that way, just based on it's very difficult to kind of figure out how much force all these things are producing. So isolating the fact that a binding site occurs, that one does in fact occur, and kind of doing this approximation would lead to further analysis. But this work needs to be done first.
Let's not stop yet. I want to ask the last question.
Yeah.
Because somebody asked you about mechanism. So you rule out calcium. Let's assume it binds. Let's assume that happens. And you activate this calcium, that you inhibit cross bridges. It's not happening for you. You get the same result. But now you allow cross bridges to attach. And all of a sudden, you get that stiffening of titin. All of a sudden, you get that extra force. So can you think about what might be happening, what might be a molecular mechanism that potentially might account for that?
I would say there's two ways to look at it. One is that it might be conformational changes. So the actual of like I think it's troponin and tropomyosin that cover the actin filament. So it might be cross bridges that form between the two that cause movement of that, or slight changes to positions that free up binding sites.
And that's exactly what happens. So when you activate myofibrillar muscle, this calcium, then the troponin tropomyosin complex moves on the actin. It frees up binding sites for cross bridges to attach. When cross bridges attach in a strongly bound state, tropomyosin moves yet again by about the same amount. What if that second movement, when cross bridges bind in a strongly bound site, what if that might potentially then free up the binding site for titin, for example? So that, I'm not saying that's what it is, but that would be the regulatory proteins that allow attachment or not attachment of cross bridges might also allow attachment or not attachment of titin potentially. I mean, that might be one of probably 10,000 mechanisms that one can think of.
I think the other likely one is that if you have cross bridges, you're bringing the filaments closer together, but you're bringing them closer together you're bringing them closer together in a state where titin is still it's not fully extended. It's still like a randomly disordered protein. It's still wavy and stuff. So it has the opportunity to extend to and then attach onto the actin. Whereas if you were to stretch a sarcomere to say 5 or 6, yeah, you're still gonna, because you're stretching it, you're gonna bring the filaments a little bit closer together geometrically. But now the titin is stretched out already, then the binding site won't happen.
So Venus, how much does the lattice spacing change at the same length when the muscle is activated and it's not activated?
I do not remember, but I know that I did experiments in residual force enhancement, and it wasn't different between the reference state and the force enhanced state, despite the fact that force was higher in the force enhanced state. So, in my opinion, it's not that.
It's not that explanation. Okay.
I think it might be, 0.1 nanometer of lattice spacing change out of 37 or 40, and so you have 0.1 out of 40 or 1 in 400, so a fraction of a percent. I'm not saying that couldn't happen, maybe very, very tiny configurational changes could do a lot of difference. But then I'm thinking when I have a sarcomere at 3.5 microns or 2.5 microns, then my lattice spacing changes by an enormous amount, by multiple nanometers. Whereas activation either doesn't do anything or maybe a fraction of a nanometer like that. I have a little bit of a hard time believing that might be an important thing, but nobody knows that.
Follow up and very naive follow up on the mechanism of titin and the cross bridges are required. So why it's not a cross bridge mechanism? That cross bridges are becoming like producing more force suddenly? Because it is related to the cross bridge. So why are you thinking about their affecting titin, actin, are the effects being
For me, the reason not to look at the cross bridges themselves is just because you do get passive force enhancement once you deactivate a residual force enhancement muscle. So you'll still have a little bit higher force for a little while. And that just means that, okay, now the cross bridges are completely out of the equation and we still have a little bit higher force. So it's probably not the cross bridges.
I would say, like, I would be careful with just making it all about Titan and excluding completely any contribution of maybe increase of force per cross bridge, because if the European was accomplished, it was the idea was that Titan, not in the I-band region that is affecting the myosin and it's interacting with, and it is interacting with active, it's also in the A-band region, it's affecting myosin and the cross bridges. So just a word of caution.
And usually the passive force enhancement doesn't really account for the entire amount of force enhancement. So it's possible, likely, that something else plays a role there as well. Sorry, Steph.
Well, let's thank Armaan again.